So today we're going to be talking about the molecular biology of the gene, and particularly about DNA structure and its replication. So in the early 1900s, it was still not clear what was the genetic material that allowed us to pass along information from one generation to the next. So there was a big push in the scientific community to try to figure that out. And so it was deemed that there were three requirements that were necessary to be considered genetic material. The first was that it had to be able to store information, obviously. Number two was that it had to be able to replicate accurately, because we can't have lots of mistakes being passed on from one generation to the next. However, number three also had to be fulfilled, and that is that there was that you had to be able to undergo rare changes or mutations so that we could provide some genetic diversity. So lots of experiments went on, and I know I've got this in this small little thing, but it was a big deal that Hershey and Chase in the 1950s pretty much finalized that DNA was the genetic material. They did the definitive experiment on that. So if you think about it, the 1950s actually wasn't that long ago. 1950s, I just think of like the typical like poodle skirts and all that kind of thing, and that is when Hershey and Chase came up with their experiment that defined DNA as the genetic material. So once it was discovered that DNA was the genetic material, then it became the goal of scientists to figure out what the structure of DNA was, and then of course after that the function, but first they wanted to just figure out the structure. And so that began with Rosalind Franklin, who figured out that the DNA was a helix. And she figured that out using X-ray diffraction techniques. And then Erwin Chargaff figured out that what holds that helix together is the pairing of bases. And in fact, we get what we call our Chargaff rules, which means that the adenine and thymine are always paired together, and the guanine and the cytosine are always paired together. And those bases coming together through hydrogen bonds hold the helix together. And then Watson and Crick took a look at everybody's work, as all scientists do. They will read all of the articles related to the subject and then come up with a hypothesis. And Watson and Crick did this, and then their hypothesis proved to be correct. And that was that the structure in its entirety was a right-handed double helix, obviously held together by those base pairs. And so they are credited with the final structure of DNA. So here's just a fun little side note for you. Crick, being one of the geniuses that he is, also did have some quirky ideas. So Crick did the calculations to see what the probability of the DNA molecules coming together on their own or self-assembling would be, because remember at the time there was the thought that there was a primordial soup where all these molecules would come together and build something. Granted, that goes against one of the laws of thermodynamics, but that was the general thought at the time. So Crick said, well, what's the likelihood that this DNA molecule would self-assemble? And when he calculated the probability of that, it was so astronomically small that he came up with a new theory, and that theory is called panspermia. And so essentially what that meant was that he believed, because it couldn't self-assemble, that aliens must have come and seeded our planet with this DNA. So just some fun factual information for you. Sometimes our genius scientists also come up with some crazy things. Another example is Jacques Cousteau, who was an absolutely wonderful marine biologist, also thought that humans could adapt and breathe underwater and was determined to make that happen for himself, but that was not the way adaptation worked. So anyway, so that's just a little bit of fun, but that is how the final structure of DNA was discovered. So then when we knew what the DNA structure was, then scientists started looking at the DNA composition across a variety of species that were already known. So pulling the DNA from various things, what does that look like? And what we discovered was that the percentage of nucleotides varies widely across species. But when we look in one particular species, the individuals have about the same nucleotide composition. And so, like for instance, in humans, they would have about the same amount of nucleotides as each other. Doesn't even matter if you're male or female, that sort of thing. And then what we also looked at was the complementary base pairs that are found in each of these species, and it reflects exactly what Chargraph had described earlier, and that is that when we look at the adenine versus thymine, those percentages are roughly the same, and so are the guanine and cytosine. So when I have up here DNA composition of various species by percent nucleotide, that percent nucleotide is based on the base that is within that nucleotide. So that's what you're looking at on this particular chart. So if we look at humans, they have about 31% adenine, 31.5% thymine. So clearly we can see how those are paired well. And for our guanine, we have 19.1 and cytosine about 18.4. So, but if we're looking strictly at adenine, we'll see that humans with only 31%, but E. coli have about 24 to 25%. So there is a variety across species, but within the same species, they would be the same. Okay, so let's review our nucleic acids. So our nucleic acids, RNA and DNA, are made up of nucleotides. And those nucleotides contain a pentose sugar, a phosphate group, and a nitrogen-containing base. And this is true for all of them. So here's our phosphate group up here. Here's our pentose sugar. In this particular one, we're looking at deoxyribose, which is DNA. If it were RNA, it would be ribose. And then here's our nitrogen-containing base, which can either be adenine, thymine, guanine, or cytosine. So here is our DNA monomer. So we have a deoxyribose, that's our sugar, our pentose sugar, our phosphate group over here, and then our base, and that base can vary, can be any of these four. Okay, and so here is RNA, and note that we have ribose here, that's our pentose sugar, again our phosphate group, and any of these four bases. Notice that this time we have uracil instead of thymine, so the four base choosings for RNA do differ from DNA. So just a real quick review of RNA versus DNA. The first being the ribose sugar that's used. In DNA, we have the deoxyribose, and then in RNA, we have ribose. Our nucleotides in DNA can be adenine, guanine, cytosine, or thymine. In RNA, we still have adenine, guanine, and cytosine, but this time we have a uracil and no thymine. DNA is double-stranded, whereas RNA is single-stranded. And DNA makes up your genes, we'll talk about what that is, and RNA is used to make proteins. Okay, so just reviewing, we're going to start looking at the structure of DNA. So these are our four nucleotides. We have two that are purines and two that are pyrimidines. So this is a bit of a review for you, but I do want you to be able to draw these structures out because they are important for how DNA functions overall. So when we look at our purines have the double ring here. And so here is our deoxyribose, our phosphate group, and for adenine, we have this double ring right here with the nitrogen right there, the amine group. For guanine, again, same base, we have our deoxyribose and our phosphate, and guanine has this double ring because it's purine, and at the top you will notice that it has carbon double bonded to oxygen. Our two pyrimidines, thymine and cytosine, again we still have our deoxyribose and our phosphate group, but these have single ring bases. So our single ring base with thymine has carbon double bonded to oxygen. And for cytosine, we have our single ring with the amine group up here. Okay, so you should be able to recognize those structures or draw them out. So our overall DNA structure has a ladder-like look to it. Obviously, it's going to be twisted into a helical shape, but if we were to flatten it out and look at it, the sides of the ladder are composed of the sugar and phosphate groups, so our deoxyribose sugar and our phosphate group, and then the rungs of the ladder, or the inside, are composed of the base pairs. So do you remember hydrogen bonding from chapter 2? When we talked about water, we said that what makes it a liquid is this kind of weak bonding called hydrogen bonding. Well, that's going to be important in our DNA structure, because our bases are held together by hydrogen bonding. And so our thymine bonds to our adenine through a double hydrogen bond. And then our guanine and cytosine has a triple bond, but these are all hydrogen bonds. So hydrogen bonds are weaker than our covalent bonds, and that's going to be important when we start replication. But this double helical structure is a very stable shape for DNA. So something else to know is that DNA strands are complementary. So if you have one strand that has the thymine, the other strand is going to have the adenine. So they complement one another, and that works all the way down the chain. When we talk about the backbone of the ladder, the outsides, those are held together with a stronger bond. That means the nucleotides are held together by a covalent bond called a phosphodiester link. And so you can see, here's the phosphate group, and it links, let me see, sorry, trying to get it right for you, right here with an ester group. And so we call that a phosphodiester linkage. And so that makes up the outside backbone of the DNA, and those are stronger bonds than the hydrogen bonds that link the inside. So what do we mean by the DNA runs in a 5' to 3' direction? Well, when we look at our DNA nucleotide, here we have the 3' section with its hydroxyl group and the 5' part of the ribose that contains a phosphate group. These are the two on the next nucleotide that are going to link together with a phosphodiester bond. And this 5' is going to link to the next nucleotide's 3', so 5' to the next 3'. Okay, so here we see it again, 5' phosphate to a 3' hydroxyl. So that's here to here. And on this other side, remember it's complementary, and it's also anti-parallel, which means that it goes in the opposite direction. So 5' here to 3' that way. So they're complementary, but they are also what we call anti-parallel because they run in two opposite directions. So here we go. 5' to 3' on one strand and 3' to 5' on the other strand. So typically what we would say is that the 5' to 3' end is called the template strand, and the 5' to 3' end in the opposite direction, right here, is the complementary strand. So this is what it would look like, particularly here. If we looked end on, we would see the bases in the middle, and then the sugars and phosphates on the outside. And here's our side view. So here in the center are all our base pairs, and then our sugar and phosphate backbones right here. And note that it is a double helix, which is a little easier to see in this image over here. So here's the one strand, and here is the other, our double-stranded DNA, making a double helix. And so when we do that, we have what's called a major groove because it has a larger spacing, and a minor groove. And for each full turn of the helix, there are 10 base pairs. And usually we will signal base pairs by BP. So in other words, if we find a particular section of DNA that's new that we say discover, we could say, hey, it's 250 base pairs long. So we would designate that as 250 BP, and that just means the base pairing. And knowing that every 10 is a complete right-handed turn of the double helix. Okay, so now you know the structure of DNA. Now let's talk about the replication of it. So this is how we pass it down to either the next generation or just in dividing and growing our cells. So let's talk about just dividing and growing our cells for right now. So all of this is going to take place in the S phase of the cell cycle. And so the way DNA is replicated is what we call the semiconservative replication. And what that means is that we're starting with our double-stranded DNA, and we're going to call these the parental strands. And so that means this one strand here and this second strand here. And we're going to split them. Notice how they are going into two right here. And then each one of those is going to serve as a template to make a new strand, which is called our daughter strands. So our daughter strand, once everything is completely replicated, will have one strand of the parental DNA and one new synthesized strand. So hence the term semi-conservative. So again, we start with two parental strands, and at the end we have two daughter cells, each of which has a single strand of parental DNA and a single newly synthesized strand to make their new double helix. So we begin this replication at what we call a replication fork. So if this road serves as the double helix DNA up here, we are going to split it like this, and it will give us two replication sites. So a replication fork. So if we think of this DNA as this long strand of double helix that we have to unwind in order to replicate, eukaryotic cells have multiple sites where they start that replication. It doesn't just start at one end and work its way to the other. To make it a little faster, it has different points that we call the origin of replication. And so at the origin of replication, we need to open up that helix and allow the DNA to be replicated. So it essentially has two sites of replication forks, as you see here. And then this creates what we call replication bubbles. So the opening of that strand at multiple sites gives us replication bubbles in order for the DNA to be replicated. Now, I am pretty sure that all of you are way too young to have used this type of phone, but it does demonstrate something pretty well. So on these old-timey phones, we had these phone cords. And before you think I'm too old, I didn't use this very much, but I do remember when I was really little having a phone that had a cord. And just a side note, there is no sound better than a corded phone on a landline. No cell phones sound as good, I just gotta say. Okay, but back to our story. So this acts sort of like the helix of our DNA, and if we were to open it up, you would notice that it starts to get very tangled because there's so much tension within these coils. And I remember that was always a problem with these kind of phone cords. But because our DNA looks similar to that, we have to take care of that problem. So there are many enzymes that come into play during the DNA replication, but we're going to highlight several of them. So the first thing I'm going to mention is what we call DNA topoisomerase. And that is the enzyme that essentially alleviates that tension, that cord being all tangled up because of our opening is going to be alleviated using this particular enzyme, DNA topoisomerase. And the molecule that unwinds the DNA is what we call the DNA helicase. And so that untwists the DNA to allow for this replication fork. Now, to make sure that it kind of stays open and stabilizes it and so that the DNA doesn't go back into its helical form, there are these single-stranded binding proteins that get put all along the way. And the reason for that is because the helical shape is so much more stable than the unwound state. So the DNA would naturally want to go right back to that back to the helices. However, these single-stranded binding proteins keep that site open so that replication can occur. So before a new DNA strand can be synthesized, a small primer is added on the parental strand, and that is called the RNA primer, and it's placed there by the RNA primase enzyme. And what that's used for is the DNA polymerase recognizes that small little portion of RNA as a target and allows it to start bringing in new nucleotides to form the complementary strand. Now, DNA is always synthesized in a forward direction, so it has directionality to it. And it starts with the 5' end right here that has the exposed phosphate group, and it synthesizes down the strand to the 3' end, which has the free hydroxyl group. So it's always in that direction, 5' to 3'. Okay, so as our double helix is unwound and we have our replication fork and our origin site, DNA polymerase adds new nucleotides to the new growing strand. So here we go in that direction, 5' to 3'. We have incoming deoxynucleosides. So DNA polymerase brings in those new nucleotides, sorry, hard to speak, and it also covalently links those nucleotides together with that phosphodiester bond. So here's our RNA primer, and then So that's also right here. And then it starts to add those nucleotides one at a time. Brings them in the complementary pairing. And then it also covalently bonds them together in this direction. So here is our replication bubble. We have the origin of replication right here down the middle, and two forks running in the opposite directions. And for every replication origin, we have a leading strand. It's going in 5' to 3' direction, and a lagging strand still in 5' to 3' direction, but it goes into these small fragments. So let's talk about that just a little bit. So if we look at the parental DNA as it's unwound, if you'll remember, our two strands are going to run anti-parallel to each other. So for the parental DNA, we have one strand here that's running 5' to 3', and the other strand is a 3' to 5'. That's the direction of the parental or the template DNA. Well, our DNA polymerase can only synthesize in the 5' to 3' direction. So when we're looking at our replication fork, keeping in mind the parental strand is running this way, so it'll have a 3' end, and our DNA polymerase can come in here and make the complementary DNA in the 5' to 3' direction. And that's our leading strand, so that one replicates a little bit faster. For the original parental DNA that runs in the 3' to 5' direction, keeping in mind again that our DNA polymerase can only work by synthesizing 5' to 3'. So it has to come in and make things in small fragments at a time, each one being 5' to 3'. And these are called Okazaki fragments, just after the scientists who found out about them. So our DNA polymerase makes one long leading strand all continuously, and it does that for the leading strand or the parental DNA that runs in the 5' to 3' direction. The Okazaki fragments are found in the lagging strand, which is complementary to the parental 3' to 5'. I know that might take a minute, so if you need to review that again, that's fine. So we have one long continuous leading strand, but then we have Okazaki fragments that then need to be connected together. So here we go, just taking another look at this again, and this is a figure right out of your textbook. So we start with our leading strand that can replicate all at once in one continuous line from 5' to 3'. That's our leading strand. Our lagging strand, the replication occurs in those fragments called Okazaki fragments. And so these two both occur using the DNA polymerase. DNA polymerase has a lot of functions, and it's irreplaceable, basically. Okay, y'all know I can't write with this pen. I hope you can tell that's DNA polymerase. Okay, after that occurs, DNA polymerase replaces the RNA primer with DNA fragments, because remember, one of the main functions of DNA polymerase is bringing in those nucleotides. So it's going to replace that RNA primer with DNA. And then another enzyme called DNA ligase is going to mend all the fragments together. So it mends the Okazaki fragments, and then it also mends where the DNA polymerase has replaced the RNA with DNA. That becomes a little fragment, and so that is bound together with the rest of the DNA strand. Okay, so let's put it all together. So to begin with, we have our helicases that unwind the parental double helix. I have added on here what was not on here, which is our topoisomerase, which remember that keeps the rest of the helices from getting all tangled. Then our single-stranded binding proteins stabilize the unwound DNA. Remember, because otherwise it would want to collapse back into the double helix. And then we have our leading strand is synthesized continuously by in the 5' to 3' direction by DNA polymerase. And actually what I don't see on here, which I should see, is there should be an RNA primer about right here. Okay, so we'll pretend there's one of those here. So RNA primase would have put an RNA primer right here. On the lagging strand, DNA is synthesized discontinuously or in fragments called Okazaki fragments. And remember, each time that we start our replication, we have RNA primers. So there are several put along here. And the DNA polymerase is acting to synthesize in the 5' to 3' direction. And then it also will come in and replace the primer with new DNA. That's a different DNA polymerase. And then the DNA ligase comes in and binds all the Okazaki fragments together, allowing for a continuous strand on the lagging side. So that is a summary of DNA replication from start to finish, starting with our double helices, coming in, the helicases will break those hydrogen bonds to unwind the DNA. Our single-stranded binding proteins will keep them apart while the DNA polymerase does its work of replicating the DNA and making a new strand. Each one of those starting points are going to have an RNA primer put in by RNA primase. On our lagging strand, we have Okazaki fragments that will be joined together by DNA ligase. And after this, we should have a parental strand and a daughter strand making the new double helices. That is the semiconservative replication of DNA. So what happens if there are mistakes along the way that are made with our DNA replication? So it's not perfect. Scientists have found that about 1 in every 100,000 nucleotides are replaced incorrectly. Well, that's actually, even though that's a small percentage, based on the amount of nucleotides that DNA replication involves, that can be quite a bit. So DNA polymerase also proofreads the new strands to check for mistakes. So this DNA polymerase does a lot of things. It's a pretty amazing enzyme. So if the mistakes are not caught by DNA polymerase, there are other proteins called DNA damage proteins that remove the bad DNA, and then those gaps are filled in by, again, DNA polymerase. So there are 3 different types of DNA mistakes that we see. So up here on the left is the correct version, our template and the complementary strand that is created. So this is what we're expecting to find if everything goes right. Well, sometimes we have a deletion, so the missing T that should be here isn't here. Occasionally there's an extra nucleotide in place, so an addition problem, or sometimes there's a base substitution. So where this should be T, an A has been added. So this is where we find mistakes in the replication of DNA. So in addition to DNA polymerase's function to synthesize DNA in the 5' to 3' direction, it also has a function called the exonuclease function, and that works in the 3' to 5' direction. And that's part of its proofreading ability, is as it looks in the 3' to 5' direction, if there is a nucleotide that's in there that should not be, it can be taken out using this exonuclease function. Yes, this is what the DNA polymerase looks like. No, you don't have to recognize it. I just wanted you to be able to see it. So DNA polymerase has multiple functions, and it helps to not only synthesize our new DNA strand, but proofread it and fix it. So what about replication in prokaryotes? So far, everything we've talked about has been in eukaryotic cells. Well, if you'll remember, prokaryotic cells have one chromosome that's tightly wound into a circle. So that's their DNA, this circular DNA. So what that means for when we begin replication is, remember in eukaryotic DNA, we had multiple sites of origin to replicate the DNA. And keep in mind that the eukaryotic DNA is much, much larger than our prokaryotic DNA. So it takes multiple points of origin to get it done in a timely manner. Our prokaryotic DNA, being a single chromosome, a single circular strand of DNA, has only one point of origin. So this is what that's going to look like. We have our site of origin right here, and two DNA polymerases will come in and work in opposite directions. And as they do, they continue to replicate until they come back together and meet each other, and that's called the termination of replication. And then the two strands are separated. So it's a little bit simpler, but all the mechanics still work the same. But that's how it differs from our eukaryotic cell. So, what was the purpose of DNA replication? Well, that purpose is for cell division, passing along that genetic information as our eukaryotic cells and prokaryotic cells grow and develop and divide. Gene expression has a different purpose, and that is to make proteins. And so the central dogma of molecular biology for gene expression is going from DNA to RNA to amino acids that make proteins. And that's going to be the topic of discussion in our next lecture. So review DNA replication and make sure you understand that and all of its players, and we will move on to gene expression next.